in F2 generation. To calculate out the results in F2 generation, let us make a square four types of alleles are present, so 16 types of gametic combinations can be made out of this. So we have to draw a table. I'll draw it again because it's four rows and four columns. Very simple. Here we have to write capital Y, capital R. Here we have to write capital Y, small r. Here we have to write small y, capital R, small y, small r. Similarly, here also capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. If we will cross self-pollinate, then the chances here can be of 16 type of genotype. You will write small capital Y, capital Y, capital R, capital R. Again capital Y, capital R, small r. Again capital Y, small y, capital R, capital R. Capital Y, small y, capital R, small r. Capital Y, capital Y, capital R, small r. Capital Y, small r, small r. Capital Y, small y, capital R, small r. Capital Y, small r, small r. In this line, capital Y, small y, capital R, capital R. Capital Y, small y, capital R, and small r, small y, capital R, small y, capital R, small r, capital Y, small y, capital R, small r, capital Y, small r, small r, small y, capital R, small r, small y, capital R, capital R. Here we should remember that wherever capital Y is present, the seed is going to be yellow. Wherever capital R is present, the seed is going to be rounded. If small y and small r is present, then it is green and wrinkled. So we can see there are 16 columns over here, 16 rows here. This is yellow seed with rounded form. Again yellow seed with rounded form. This is yellow seed with rounded form. This is yellow seed with rounded form. Yellow, rounded form, yellow. Now because of this small r, it will be wrinkled. Yellow wrinkled. Again yellow rounded. This is yellow and wrinkled because of presence of small r. This is yellow rounded. This is yellow rounded. This is green because of presence of small y. This is green rounded. This is green rounded. Again this is yellow rounded. This is yellow, but some small r makes it wrinkled. So it is wrinkled. It is yellow, not green. Sorry, it is green, not yellow, because of presence of small y. This is green and rounded. This is green and wrinkled. If we see, there are in all 9 plants with yellow and round form, 3 plants with yellow and wrinkled form, and 3 plants with green and rounded form. Oh, sorry, green wrinkled form, and only one in the form of green rounded form. So the ratio of genotype as well as phenotype in Mendel's dihybrid cross is 9 is to 3 is to 3 is to 1, where 9 stands for yellow and rounded seeds, 3 stands for green and rounded seed, again 3 stands for yellow but wrinkled seeds, and 1 stands for green wrinkled seeds, which is this one. So these are the results of 2 crosses conducted by Mandel. One is known as monohybrid cross, another is known as dihybrid cross. Similarly, Mandel conducted trihybrid cross also, where we need to have a broader square to be drawn, but the method will remain same while calculating the trihybrid cross. The rules will be same. After doing these 2 interesting experiments, Mandel reached to some outcome conclusions. They are studies at Mendel's law of inheritance. What were those laws? One is law of dominance. Law of dominance says that the characters which are expressed in F1 generation are dominant characters. This law can be repeated as when the 2 homozygous individuals with one or more set of contrasting characters are crossed, then the characters that appear in F1 generation are dominant characters, and the characters which are not expressed in F1 generation are recessive characters. So this is law of dominance. This was based on Mendel's monohybrid cross. Second is the principle of segregation or law of segregation. Law of segregation says it can be also written as law of purity of gametes. Law of purity of gametes or law of segregation is that when a pair of contrasting factors or genes are brought together in a heterozygous individual, the 2 remains together without being contaminated. The 2 alleles remain together without contamination. Means there is no mixing. But when the gametes are formed from them, then the 2 alleles separate. For example, capital T and capital T will remain together. But if meiosis takes place in this cell, then they will separate out. That is law of segregation. It is also known as second law. It is also known as second law of heredity. Next is principle of independent assortment. Independent assortment. Independent means freely. Assort means to reunite. If the inheritance of more than one pair of characters is studied parallelly or simultaneously, the genes for each character assort out independently. This law says It is called Mandel's second law also, but it is not second, it is third law basically. But commonly it is also known as second law. Independent assortment can be studied by the example which was just discussed over here, that if we cross capital T with small t, then this is free to assort with any of the allele. It may join to this one, it may join to this one, this may join to this one, and this may join to this one. Means any allele is free to join with any allele. This is known as independent assortment. But this was drawn out of Mendel's dihybrid cross, where he studies two contrasting characters at a time. By these laws, we can confidently say that genes are hereditary units which are responsible for transmission of characters from one generation to the next. Genes are hereditary units made up of DNA, responsible for transmission of characters from one generation to the next. With this, we conclude this session. We'll discuss remaining part in the next time.